right, well, happy Monday. Hope the weekend was good to you. I know this week we're looking forward to some beautiful weather, so hope we can get out and enjoy it. I think it's going to be like the 60s at one point, so it's nice. Not really typical for November, but we'll take it, we'll enjoy it. I want to make one announcement. So I know we're running a little behind in lecture, right, and that is okay. The test is on Monday. The last material that'll be tested on will be presented on Wednesday. So even if you don't get to all of the topics listed on the schedule on Moodle, that is okay. Whatever we get through on Wednesday, that'll be it. Right? Just so you're aware of that. Any questions to start off class? Yeah. Not in person, but if you still have questions, let me know. I'm going to start off class with reviewing the class question we left off with on Friday, which is similar to that content check. Yes. Any other questions? All right, so we did leave off on that content, or not the content check, the in-class question that we did on Friday. You had some time to work through this. And so now I have provided the answers to you. You should see that on these slides that you have, right? And hopefully you figured out, right? This is our template strand. We have to make that mRNA. So transcribing it to mRNA, translating it to our amino acids, right? And then figuring out how these different mutations would affect our translation. All right, so hopefully you got to the conclusion that the first one here was a missense mutation. All right, so we changed out the amino acid in this first amino acid codon here. That second one you should have found was a silent mutation. So even though a mutation occurred, we did not change the amino acid. All right, same for that third one here, also a silent mutation. And that last one was a frame shift mutation. So we inserted in that additional base pair there, and we got a frame shift mutation. So the entire frame after that mutation changed how we did it. Any questions on that? Right, I can guarantee that a question in this vein of thought will show up on the exam in some format. So make sure you take the time to understand this and practice it. I am planning on putting up some practice questions up on Moodle, hopefully tomorrow. That is my plan. Tomorrow, if not Wednesday. All right, so we're going to finish up our talk of mutations today. We talked about mutations in the gene, right? But there could also be mutations outside of that DNA sequence that encodes for our gene. Remember, we have those sequences like the promoter or the terminator sequence. Right? We also have those introns. And so if a mutation occurs, say in the promoter region, right, that region right before the gene, we might affect the rate at which transcriptions would happen. Any of those transcription elements that might regulate transcription, if we have a mutation in those areas, we might see this being expressed as, we might alter how much of a gene is being expressed or when it might be expressed. Those splice sites, remember we're splicing out our introns here when we are processing our RNA in materials. If we have a mutation in those splice sites, it might affect how we are properly splicing our mature RNA. And any elements that might regulate translation. The mutation occurs there, we're just going to mutate and have a mutation in our ability to translate our RNA. Right? So big picture here, right? Mutations can occur within a gene, but it can also occur outside of a gene. Right? So our DNA sequence, we can have a mutation outside of the sequence that encodes for the genes, and we can have various results depending on where that mutation occurs. All right, we can also look at mutations in terms of where during cell division a mutation might arise. And so if we look at germline cells, right, so these are cells that are going to give rise to gametes. So think egg and sperm in humans and in animals. Right? If we have have a mutation in the gamete, so directly in the egg or sperm, or any cell that might be a precursor to those gametes, so the cells that go through meiosis to produce the gametes. We could get these mutations that would affect the entire individual. Right? So, in this example, we have a mutation in the sperm. Right? Fertilization is occurring. We are creating a zygote that turns into an embryo here. That embryo, those cells are going to go through mitosis. Okay? And every one of those cells is going to have that mutation because that mutation was originally present in the gamete. Okay? And this could result in some of the offspring potentially carrying that mutation as well. Right? So, mutation in the gametes can lead to mutations in the entire mature individual. Right? But we can also have these mutations in somatic cells. So cells that are not gamete, cells that are not egg and sperm. And so an example up here is we have a mutation in our embryo here, right? And it's this mutation that would kind of be like the shoulder, precursor to the shoulder area of a mature individual. Okay? This mutation can happen either late or early in embryonic development, right? So it's happening after fertilization has occurred. And as that individual grows up, we might see a patch of the affected area having that mutation, rather than the entire individual. Right? And so when this individual reproduces, right, their gametes are not going to carry that mutation. Right? A good example of this, if you've seen individuals that might have black hair, right, but have a patch of white hair, right, that's a good example of a somatic cell mutation. Right? And that's an example of something that's easily visible for us to see. Right? So this might not be super obvious, right? But in the hair follicles, you see a patch of white hair, that could be an example of a more obvious somatic cell mutation. All right, so we talked about mutations in the gene. We talked about mutations outside of the gene. We talked about where mutations can occur in cell division. Right? But we haven't really talked about the cause of mutation. So what actually causes a mutation? Right? Well, there are several things. So mutations, they can be spontaneous. Right? They can happen in some due to some biological cause. Right? So it could be an error in DNA replication. Right? So maybe DNA polymerase is putting in the wrong base pair. And we know it has the ability to proofread, but maybe one got past that proofreading ability. Or it could be other byproducts made by the body that are not properly being disposed of or regulated that could interfere with the DNA in these individuals that might have a mutation. Right? So they can be very spontaneous. Remember, these mutations are random. Right? They're not being, individual is not causing a mutation to occur. Right? It's just a random mutation. They can be spontaneous biologically, or they can be induced. All right, so an induced mutation is going to be a mutation due to some environmental element. Right? So that environmental element could be chemical in nature. So a really good example of this, cigarette smoke here contains a chemical that will actually jam itself into the DNA of individuals, and that can cause mutations and like delete some of those base pairs. Right? So chemical in nature, like cigarette smoke, certain preservatives, right? Different types of chemicals you might find in other products. Right? It can also be physical, right? So a physical breaking of DNA strand or physical deletion of some base pairs. Right? Good examples of this would be like UV radiation from the sun, which is why you often want to wear sunscreen if you're going to go out in the sun, because that could eventually lead to skin cancer, right? Because of this radiation, this physical effect on our DNA. Right? And also X-rays, right? Which is why if you ever had an X-ray done, they might put that covering over the rest of your body to prevent any X-rays from causing damage. There can also be different infectious agents that can have an effect on the DNA as well. Right? So there's a lot of causes of mutations out there. Right? I want us to think of a question now. Right? I want us to think of why might scientists intentionally cause mutations into organisms. Right? And I want you to frame your thinking in terms of crops. Right? So why might a scientist intentionally induce a mutation in a plant that's used as a crop? I'm going to give you a couple minutes to think about this. Talk to your neighbor, chat it out, and I'm going to ask you to share your answers. All right, I'm hearing some really great discussion here. So, what do you think? Why might scientists intentionally cause a mutation, specifically in crops? What are some of your ideas? Yeah, maybe we're making them more drought resistant or bacteria resistant. Oh yeah, we could breed for more like cold or heat tolerance. Yeah. What's some other examples we thought of? I was just thinking like seedless watermelons and things like that. Like interesting mutations to sell them. Yeah, increasing crop viability, or maybe we're changing the fruit to meet our needs. All right, so watermelon, they don't have to be seedless, but it's easier to eat if they're seedless. Yeah, these are all some really great examples. And so scientists will often intentionally induce mutations to try to find some sort of beneficial mutation that might occur. So in the example I have up here, right, we are inducing mutations in several different ways here to find a more disease-resistant variety of crop. Right? We can increase the crop yield. That can help kind of decrease food scarcity if we're boosting our crop production. Right? There's different ways of inducing mutations, or different ways of breeding plants to see these mutations arise. Right? So one would be simply breeding them together. So we might breed a variety that is susceptible to disease, right? Or a variety that is resistant to disease. Right? And over time, future generations might be more disease resistant. We could induce those mutations via chemicals or UV radiation. Right? We could grow up the seeds of that individual where the mutations were induced, grow a whole host of plants and then try to select out the plants that seem to be more disease resistant. There's also a process where we can intentionally insert a gene that is more disease resistant into our crops to get these disease resistant plants. This is a method known as genetically modifying our organisms, so if you've heard of GMOs. And a newer way of doing this is using a method where you can directly go in and target the gene you want to alter and engineer it to be more disease susceptible. There's multiple ways of doing this. This is a very common way of doing it. When I worked at the U of M for a while, I worked in a lab that was trying to find plants that were more heat tolerant in these methods that I described here. And it's a very common way of trying to find some beneficial mutations. Are there any questions? All right. We're going to move along here. All right, so we have these mutations occurring. How might our DNA be repaired? Right? So sometimes mutations occur, but the cell can see these mutations and correct the mutation before it ends up being passed down to other cells, through mitosis or meiosis. And there's common types of DNA repair. Right? So DNA can be directly repaired. Right? So as we are synthesizing DNA, there are some repair enzymes that can recognize that a mistake has occurred and can go in and fix that mistake. Right? There's also a method where, again, we are detecting that mistake has occurred, and we can go in and cut out that mistake. Right? Whether it's a base pair mistake or a section of base pairs, we can cut it out and resynthesize the template strands. Whether that's a mistake due to an abnormal nucleotide or a mismatch of a nucleotide, so you put in the wrong base pair. And a good example of this, if we're looking at this nucleotide excision repair here, in our top DNA strand, right, we have this abnormal nucleotide going on here. Right? This is called a thymine dimer. So instead of these thymines, binding with their with their pair here, with adenine, right, they're actually covalently bonded to each other, right, which does not allow them to form these hydrogen bonds with adenine. Right? And so because they're covalently bonded together, they're abnormal. We have to, the cell will recognize this, right, and create this bubble. Right? You can see it kind of took care of some of these hydrogen bonds here to create this bubble. We have enzymes coming in and removing this piece of DNA that was affected, and then DNA polymerase can come in and synthesize a new strand that is correct. Right, so this is a good example of how this can be repaired. Are there any questions on mutations, DNA repair, anything pertaining to the last few lectures? That's a good question. I'm going to assume it's the structure of the molecule that makes it more susceptible to that. There might be other dynamics. I would have to do some research on that. Good question. Any other questions? We're about to completely change gears here. So if you have any questions related to mutation, now is the time. Yes. Yes, we gotta see this complementary to that. Yes, just like where we saw it. Yes, exactly. It's ligase coming in and kind of sealing up those gaps and moving signals. Yeah. All right. Well, we are going to change gears to our next topic here. Ingredient inheritance. So I know we started talking about this in the lab last week, So some of this might be familiar. All right, so theory of heredity. Right? We know that our chromosomes store information in our DNA, right? Or our DNA is storing information and it's tightly wound into our chromosomes. You know this is common for all living things. Right? All living things have DNA. Right? This DNA is packaged and it can be passed down to its offspring. And we saw this when we talked about meiosis. If we look at the DNA that's passed down, right, any characteristic of an individual, we would call it the trait. The trait of the individual. Trait is what's being caused by that gene expression. So genes are being expressed, that leads to the trait that it has. Right. And that traits are passed down. Right? And you see this being passed down from parents to offspring. So this would be So we're thinking about how traits are being passed down. And this is what Gregor Mendel was thinking about when he started his experiments. Right? He wanted to know, how are these traits passed down? And so he did a series of experiments in the 1860s on pea plants, right? And led to a bunch of different discoveries of heredity and inheritance. Kind of some fun historical facts, right? So Gregor Mendel, right, was around and alive and publishing work the same time that Charles Darwin was, right? But they were actually unaware of each other and each other's work. Right? So kind of a fun fact. Also, Mendel's work was actually lost. Right? His work was lost and was rediscovered in the 1900s. So, interesting, fun historical facts about Gregor Mendel and his work there. So Mendel was really trying to get at the root cause of these two basic questions. Right? Why do offspring resemble their parents? I know when I was growing up, everyone would say, oh, you look so much like your mom. Right? So I know I resemble my parents. I'm sure you resemble your parents in some manner. So why do offspring resemble their parents? And how does this transmission of traits occur? Right? So how do these traits get passed down? All right, so like I mentioned, Gregor Mendel, he was looking at pea plants, or he was experimenting with pea plants. Now, pea plants happen to be a really, really great model organism because they have these traits that are easy to work with, right? So first of all, pea plants, they're easy to grow, right? They don't require any type of special care, right? They're pretty easy to grow. Their reproductive cycle is really short. So it only takes about 10 to 14 weeks for a pea plant that was planted in the ground to flower and reproduce new seeds. Okay. Think about it, he was working on a lot of fruit trees. Fruit trees take like upwards to 4 or 5 years before they can reproduce. Okay. And I'm thinking about getting seeds from that tree and planting them and then waiting another 4 to 5 years. Right? Those are not super easy to work with. But pea plants, super short reproductive cycle. It also produces a large number of seeds, so you can get a large sample size. The traits of a pea plant are really easy to recognize. Right? So for example, flower color. It's really easy to see a purple flower versus a white flower. And its mating is really easy to control. Right? So you're able to control mating of pea plants using this method of pollen transfer. So oftentimes you do something kind of like a paintbrush. Right? You essentially get some pollen from one plant and transfer it to another plant here. Right? Artificial pollination. Right? So you could directly control who was the parent for each cross that you did. Right? And when you grew up the offspring, right, the offspring will often be denoted as the F1 generation. That stands for first filial. Right? It's actually a derivative of a Latin word meaning son or daughter. So we'll often see this as F1, F2, F3, and so on. Okay. Mendel looked at a bunch of traits in his piece. Right? He looked at 7 of them in all. Right? He looked at anything from the seed shape and color to where the flowers were positioned on the plant. Okay? And these observable features that we see here, so whether it's round or wrinkled or purple or white, right, those observable features are its phenotype, right, so what we're directly observing. And in his pea plants here, we see that for each of these traits that he studied, there were two distinctive phenotypes. Okay? So some traits in other organisms can have more than two phenotypes. But Mendel kind of got lucky in that his pea plants only had two. Okay? So either yellow or green pod colors, or tall or short in stem height. Right? So Mendel worked with what we call pure lines, or true breeding. Right? So you might recognize these terms from lab last week. Right? A true breeding plant is one that when they are self-pollinated, or the plant uses the pollen that it itself creates to pollinate itself, right, they are going to end up with genetically identical offspring. I should say identical offspring, right? So they look the same here. He worked with these true breeding lines, and for example, over here, we have these male parents that produce all round seeds, and the female parent that produces all of these wrinkled seeds. Okay? He then mated these true lines together to create hybrids. Okay? And so as hybrid here, he mated these true pure lines here together to create our hybrid. Okay? And he did this for a variety of different traits. So not just pea shape, but all those traits that we discussed on that last slide. Okay? And Mendel started to notice something pretty interesting about all these processes that he did. Okay? So, first of all, right, he created these hybrids. And when he created these hybrids from these true breeding pairs, right, he noticed that there would often be a dominant phenotype. Right? So the dominant phenotype, or what we're seeing, right, masks the expression of a recessive phenotype. So notice, right, we're seeing all round seeds here, even though we crossed an individual with round seeds and with wrinkled seeds. Right? So we notice the dominant phenotype would always show up in this F1 generation. He also noticed, right, if he self-fertilized the F1 generation together, right, so we took two individuals from this F1 generation, and, excuse me, not two individuals, just one, because we're self-fertilizing, right? One individual self-fertilized the resulting F2 generation, right, have this very distinctive ratio to it. Right? So first of all, we see that we have our round peas, but we're also now seeing those wrinkled peas as well. So the recessive gene has reemerged, and it's in this 3-to-1 ratio. Right? Or 3/4 of his plant was round, or of his seeds were round, and 1/4 was wrinkled. Right? So he's noticing this 3-to-1 ratio, and not just in the seed shape, right, but in all of the traits that he looked at. He is noticing a 3-to-1 ratio going on. Right? So he's noticing this distinctive ratio. Right? He's also noticing these dominant and recessive genes. Right? So this should be a kind of review because we went over this in lab last week. So a dominant trait is the trait that will always be displayed if that allele is present. Right? So in pea plants, examples of this would be tall stems or purple flowers. Right? So if there is an allele that calls for that dominant trait, it will always be the one expressed. But we also have these recessive traits. Right? And we can see that the recessive traits are always going to be masked by the dominant trait if we have one of each allele. The only way the recessive trait can show up as the recessive phenotype is if both alleles are recessive in that individual. Right? So a good example in pea plants would be dwarf stems or white flowers. Right? And again, that dominant phenotype is hiding the expression of the recessive. And in order for this recessive phenotype to show up, we have to have both alleles be recessive. There cannot be any dominant allele. All right, so Mendel, when he did this work, right? The term gene didn't exist, right? Mendel kind of pioneered a lot of work in proteins, right? And he kind of figured out these hereditary determinants for traits were called a gene. Right? So we've talked about genes. We've also talked about alleles, right? So Mendel proposed that for a gene, right, each individual can have two versions of that gene, right? So essentially one coming from mom and one coming from dad, which would be called the alleles, right? Or the gene variants. Okay? And that different alleles are responsible for the variation in those pea plants that Mendel saw. If we look at another example that's not a pea plant, right? So for example, fruit flies here, okay? We see we have a homologous pair, right? So chromosomes containing the same type of genetic information. We see it has the gene for eye color on both of them, right? And for each gene, there's an allele, right? So the allele for the red eye or the allele for the purple eye. In fruit flies, it's a bit more complicated. There's not just two phenotypes, right? I think fruit flies have 7 phenotypes for eye color. But this example, right, because we can only get one chromosome from mom, one chromosome from dad, our two alleles are red and purple. And red eyes are always going to be dominant. So this individual would show up as having red eyes in the fruit fly. Okay? Same principles as the pea plants that Mendel proposed. All right, so those alleles found in an individual are called its genotype. Okay, so genotype refers to those alleles. The genotype is going to affect the phenotype, or what we physically see. Okay, and the location on a gene, or of a gene, on a chromosome would be called the locus. Okay, so let's use some images to kind of help us wrap our heads around this here. So we have a pair of homologous chromosomes. You can see we have a gene here for the height of a plant. Okay. You have a tall allele and we have a dwarf allele. Okay. Those alleles are determining its genotype of being big T and little t. Right? So the dominant trait, or the dominant genotype, or the dominant allele is always going to be uppercase. Right? The recessive allele is always going to be lowercase. So we have our genotype. Okay. The genotype is going to determine what we physically see, or the phenotype. So in this case, we're talking about height of the plant. You can see we could have a tall plant or a small plant, right? Or a dwarf plant. That would be the phenotype. Okay. Now in this individual, we see the genotype is big T, little t, so it has that dominant allele. And because there's a dominant allele, it's always going to mask that recessive allele. So this genotype would lead to the phenotype of this being a tall plant. All right, I talked quite a bit there, so let's take some time to think about what we just learned. This should be relatively review from lab last week, so it shouldn't take too long. But I want you, using this image of a purple flower and a white flower, I want you to figure out the phenotype and the possible genotypes for these plants in terms of flower color. Okay? I want you to use the letter P to denote those. So I'll give you about a minute or so here to work through this. How's it going, Hannah? All right, so let's start with phenotype here. What is the phenotype of this top flower? Purple, yeah, that's what we're seeing. What about the phenotype for this bottom flower? White, yes, phenotype, nice and easy here. What about the genotype for this top flower? What would the genotype be? I hear big P, yes. What else could it be? Big P, little p, yes. We don't know for sure, right? But we know it could be either of these because it has that dominant allele of the purple flower. What about the genotype for the recessive flower? Yeah, little p. All right, so when we're looking at these genotypes, and we'll use that example that we just worked through here, you could say that an individual could be homozygous or heterozygous. Right? Homozygous means it has the same alleles. Right? So they might both be dominant, they might both be recessive. So in our example here, our homozygous dominant would be this big P, or the homozygous recessive could be the little p. Right? Or the individual could be heterozygous, have one of each. Right? So a dominant and a recessive allele. We see that one of the possible genotypes for this dominant flower would be a heterozygous individual. All right, so now we get to our favorites here, Punnett squares. You either love or you hate Punnett squares. I love Punnett squares. And I know we did this a lot in lab, so we're going to kind of go through this relatively quickly. And so we're going to start with this cross between two homozygotes. Right? We see we have homozygous dominant, so big Y, and homozygous recessive, little y. If we match those all up, right, we see all of our individuals here are going to be heterozygous, and they're going to show up as yellow, because yellow is that dominant allele. Right? So 100% of those offspring are heterozygous for the yellow color. Right? And if we kind of look back to why we would set up the Punnett square this way, how it leads to these offspring being the way they are, right? So if we look at this individual that's homozygous dominant, right? So big Y, the only way that it can split up those gametes, or split up those alleles in its gametes during meiosis, is each gamete would have a big Y. Right? So only a big Y could be passed along. Same for this other individual here, the homozygous recessive individual. The only gametes that could be created would be gametes with the little y. Thus, whenever we have fertilization, it's always going to end up in this big Y and little y combination. All right. Now, what happens if we do a cross between two heterozygotes? Right, so basically we're crossing the offspring from that last Punnett square that we just did. Right. We see we get a variety of phenotypes and genotypes. Right. And looking at our Punnett square, we can figure out the phenotypic ratio. Right, so the ratio of that we're seeing in its phenotype. So how many yellow P's do we have here? 3. Yes, very good. 3 P's, and what about the green one? 1. Yes, so 3/4 are yellow, 1/4 are green. This would be a 3-to-1 phenotypic ratio. We could do the same exact idea, but except looking at the genotype, right? And if we look at the genotype, we're looking at all instances of unique genotypes. So we have big Y, little y, and 2 heterozygotes. Right? So if we looked at the ratio, we would see 1/4 of them are homozygous dominant, 1/4 is homozygous recessive, right? And 1/2 of them are heterozygous. So we would end up with a 1-to-2-to-1 ratio. Right? That ratio should sound familiar, because that's the ratio you did with chi-square tests in lab. Right? Well, why does this happen this way? I'm going to leave that as a little bit of a cliffhanger for now, right? Because I want us to make sure we really understand heterozygous, homozygous, how to assign different alleles in each generation. I want you to work through this problem. All right, so using our flowers to the right, right,
I want you to figure out what the genotypes are for the true breeding generation, the parent generation, for the F1 generation, and the F2 generation, right? And when you get to the F2 generation, think about what the ratio would be for the phenotypic ratio and the genotypic ratio. And I included a Punnett square down the bottom. You need to use a Punnett square to figure it out. So let's take about 3 minutes to work this out using the letters P for, like, the pink color is the dominant color here. So he had to kind of sort through a bunch of plants of time. This is what he did. This is what his passion was. All right, so let's look at our flower generations on the right here. What are the genotypes of my true breeding pairs? What's this first one? Big P, Big P. What's the second one? Oh, I put it up on the screen prematurely. Little P, Little P. What about the F1 generation? What is this genotype? Yeah, all heterozygous. Big P, Little P. What about the generation on the bottom? What are the possible genotypes here? Big P, Big P. Big P, Little P. Little P, Little P. Which one would be the Little P, Little P? The white one, yes. So you can use a Punnett square here to kind of figure that out and assign the genotypes if you need to. If we look at the phenotypic ratio of pink to white flowers, what would that ratio be up here? 3:1, yep, same that Mendel saw for all of his plant traits. What about the genotypic ratio? Yeah, 1 to 2 to 1. Very nice. Any questions on this? All right. Here's my answers. There you go. So Mendel, right, he did all of this work and he created these two principles that we had gone over when we talked about meiosis, right? So he created, or he came up with the, almost time, I'm going to keep an eye on the clock, independent assortment and principle of segregation, right? So independent assortment here is the behavior of those chromosomes during meiosis and the way that they randomly align on that metaphase plate. Right? So remember we have those tetrads in meiosis 1. Those tetrads are lining up on the metaphase plate, and that way that they line up is completely random, and we can get these different assortments of how they line up. Right? And so if we look at these two examples here, right, so we have this cell, 2n equals 4, so 2 homologous pairs, 4 total chromosomes, going through meiosis. At the metaphase plate in metaphase 1, they can line up in this orientation, or they could line up in this orientation, right, completely random, but the way that they line up will have an effect on the way that the gametes are sorted out in those 4 daughter cells. Right? So Mendel figured out this principle of independent assortment. Right? He also figured out this principle of segregation. Right? So the way that these two alleles separate out during gamete formation. Right? So again, we have these homologous pairs. The homologous pairs will separate, and we have all these different possible gametes that can be created. Right? We know this happens during anaphase, anaphase 1 in meiosis here. And we can also see this playing out when we do a Punnett square. Right? So we can see the gametes, or the maternal egg donor, creating these gametes. Right? And these gametes can have different possible combinations when paired together during fertilization. Right? And so Mendel figured all of this out using that 3:1 ratio of dominant to recessive alleles. And he was able to find those two main principles that we talked about so many times in meiosis, in the lab protein cell division, last week in class, and this lecture. All right, so what that's telling you is that I've talked about so many times that this will probably show up on an exam. So make sure we understand these principles. All right, I actually did end on time. So homework for Monday, all right? The content check number 8 is due, right? Remember that study guide is up on Moodle, and that scientific literacy assignment, right, will be due on Friday with the exam next Monday. All right? I am going to hold an extra study session. I will let you know what time that will be on Friday.